Hey guys, welcome back. I'm super happy to be back. I have an excellent podcast here. Two of my guests, Dr. Philip Blair, medical doctor, retired Army colonel, West Point graduate, and expert on all things beta caryophyllene, joins us for the conversation, as does Harvard undergrad in medicinal biochemistry, Stanford Medical School, Dr. Karyamet Alif, talking about the endocannabinoid systems and what are the flaws in cannabis research. We took a wrong turn a while back and haven't turned around, so it'll be interesting to hear what Dr. Alif has to say on that. How is the endocannabinoid system rewriting and rediscovering our understanding of physiology? We go into a deep aspect of that. I have a pretty cool piece where I talk about the evolution of the endocannabinoid system. And we talk also about adaptogenic herbs as well as some nutritional information. So I hope you enjoy this. It's really technical. If you're not a technical person, I think you'll still learn a lot. Just pretend you don't understand the words you don't understand, and the rest of the meaning should come through. Thanks again. Hit that like, hit that subscribe. I'm Dr. Jake Felice, bringing you these videos with a lot of love. And here we go with Dr. Alif and Dr. Philip Blair. Stay tuned.
So I'm Dr. Philip Blair. I'm an MD, and I am residency trained in family medicine. And I practiced in the Army for a lot of years, and then I retired and then went into disease management. And more recently, in the last 6 years, I've been working in the area of cannabinoid medicine and exploring so many of the different facets of cannabis as well as endocannabinoid system. And it's like being in a candy store. There's so many interesting connections that has rewrites and rediscovers physiology and all the disease entities. So it's an exciting area to be in.
Thank you.
All right. So should I go next?
Yes.
Okay. So my name is Dr. Karyamet Alif, and I'm also a medical doctor. I trained in medicinal chemistry, medicinal organic chemistry as an undergrad at Harvard, and then went on to Stanford, where I was in the cancer biology MD PhD program, and also did research in intensive care, anesthesia and intensive care, and also a fellowship in clinical pharmacology. And so from that perspective, my greatest interest really in the cannabis field has to do with, as Philip was mentioning quite correctly, the new breakthrough and insight into what's actually going on in the human body that the endocannabinoid system brings. And then also understanding, so that's the physiology side, and then understanding the pharmacology, how we impact that system with different agents, some of which come from cannabis and some of which are either stimulants of production or affect, I'll say, the production of endocannabinoid molecules within the body's own, cannabinoid-like molecules, but also to really understand what's going on with cannabis, because there's some flaws in the cannabis research that maybe we'll have time to go into, but there are some flaws that then have impacted how we perceive the endocannabinoid system and all of the associated pharmacology and effects. But as Philip was mentioning, the concept of pathophysiology, when you put the endocannabinoid system in, it's like, suddenly having the internet or a supercomputer to help you understand what's going on in the world, or, however you might describe that. Suddenly you have this much, much bigger view, and the interconnectedness of things starts to jump out at you. So that's what excites me.
Excellent. Thank you. I'm Jake Felice,
Sure.
Dr. Jake Felice. I'm a naturopathic doctor. Did my undergrad at Penn State, focusing on psychology, and my naturopathic degree at Bastyr University. I'm a plant guy. I always have been, probably since the second grade. And it's just a pleasure to see, with advances in science, how pharmacognosy and plant pharmacology has kind of come full circle, and the endocannabinoid system is smack dab in the middle of that. Cannabis is, I'd have to say, probably my personal favorite botanical, and I have just a wide ranging interest in botanicals in general. And I also come from the perspective of I'm a trained body worker. So some of the time you will hear me talk about the connective tissue or the interstitial system. I have a particular interest in those areas as well. So I'm happy to be here with you gents. We have met a few times before, and each time I've always thought it would be really great for us to record. I'm interested in what Karimet said about problems with cannabis research. I'd like to chat about that, but Philip mentioned something to me the other day about a nuclear receptor, the PPAR gamma, or one of the other ones. So why don't I hand that off to you, Philip, and just let's hear kind of what's going on with that, and what interests you about it, and how you see cannabis, cannabinoids, and other perhaps plants or compounds affecting that receptor system.
Well, the nuclear receptors, the PPAR receptors, seem to produce a cascade of different events that affect our metabolism. They're also signaling molecules and detection molecules, and they're orchestrating our lipids, and signaling and epigenetic type of changes that go on. And there's a cascade of immunologic as well as metabolic programming that occurs by manipulating or modulating these particular receptor systems. And so I've been really fascinated with the interconnectedness of those and how they broaden out to immunity and metabolism, and also how other lipids connect up with them. For instance, it turns out that these particular PPAR receptors are also receptors for many of the fatty acids, whether those are omega-3 or they are LDL or oxidized LDL. In fact, one of the PPAR receptors, a PPAR delta receptor, is in fact a receptor for LDL, and oxidized LDL. So you start to see, well, this is the connectivity that we have, and the ramifications, as well as the epigenetic types of changes, where we're making RNA modulation and we're changing our patterns on the basis of these particular molecules. I think it's quite fascinating, and we're dealing much more broadly than just the canonical endocannabinoid system. We're talking about many of the lipids that we're coming in contact with.
Wonderful.
Can I pop one little thing in there? So, I confess I'm somewhat of a purist when it comes to science and scientific method, although not necessarily in the way that people typically see that. But that said, so what Philip is mentioning about the canonical and the cannabinoid system, and also the activity of lipids, that's part of what I refer to when I mention the flaws in cannabis research. So one of those flaws, from my background experience in medicinal chemistry, if you look back into, let's say, the 70s and 80s, when much of this understanding of drug design and molecular pharmacology began to come of age, there was a very, very heavy focus on primarily three classes of compounds. Obviously, nucleic acids, so looking at DNA and the effect of DNA on, being the source code of the human body. And that was supposed to be the key to everything long before the genome was sequenced. The other thing was, of course, the associated products of DNA through mRNA, which are proteins. And so everybody was fascinated with proteins long before they were able to, sequence them so easily as now, much less have three-dimensional pictures of that site. But when it came to drugs, when it came to active compounds, whether they were natural source, natural products, or synthetics from pharmaceutical research and pharmaceutical companies, a lot of the focus was on alkaloids, on nitrogen-based compounds. The chemistry is fairly straightforward. Handling them is, I won't say easy, but was, let's say, more accepted. And so, but I mention that because in that context, the concept of having bioactive lipids was ridiculed because lipids were just seen as nutritional compounds that supply calories. The other side of that, kind of discarded coin was polysaccharides. And so again, starches, polysaccharides, sugars, they were also seen as being, they don't have the elegance and the structure of any of the compounds that I mentioned before, and really they're just there for nutrition and minor functions, structural functions, but not really pharmacologic effects, even though it was known that cell surfaces are covered with polysaccharides and oligosaccharides. But it took a very long time for those classes of compounds to even be given a seat at the table of consideration. So then when you consider what the canonical, endocannabinoid system is, part of what I believe still needs to be cleared out are those biases against these other classes of compounds. And so it's just starting to be accepted that you can have something so simple as a long chain fatty acid with a minor molecular group adjustment, like you see with the endocannabinoids or PEA or OEA, that those can actually have pharmacologic effects in the same kind of dose range or concentration range as, a regular drug or as a neurotransmitter or hormone. And so that will help also to further expand what is the endocannabinoid system really, because it was discovered because of the plant and some fundamental flaws in what people were willing to consider then drove the research in a particular direction.
That's good. And I completely agree with you, Karimet, that we really have underserved those particular areas. Now, when you're talking long chain, are you talking about 18 carbon?
Yeah, so primarily we're looking at arachidonic acid derivatives. And so, in the physiological pathways, they branch out, and in one direction you have synthesis of endocannabinoids, and in the other direction we have prostaglandin synthesis. And of course, prostaglandins, again, in that time period of 70s and 80s, those were kind of like new players coming to the table to understand inflammation beyond just simple clinical signs of calor, dolor, rubor, basis, to actually understand molecular basis of inflammation.
Yeah, but the body really likes those 18 carbons, and you see it incorporated into so many different molecules as effector molecules, whether that's the omega-3s or it's arachidonic acid. And there's such a diversity of how these things play within the different signaling systems of the body. And that sort of leads to that thought that when we confine ourselves canonically, we can to the endocannabinoid system, it's too narrow. We've got to include those other targets, as it's been called a microbiome that is inclusive. And I like to think of it that way, because then that incorporates the targets. It's like, this cannabinoid connects with this particular PPAR receptor, so we need to include this in our thinking. And then by doing that, we really extend the system. Things like ketones from fasting, they signal the PPAR receptors as well. And so you get this crossover and an integration from all of these aspects. And very well stated, Calumet, about the diversity and how we're limiting ourselves and we're not getting the full picture.
Yeah, it's if I can, I mean, since we're kind of in that little space, I'll see if I can just say it in a few words, because to me, it's like a big Disneyland of potentials. But so here we have these multiple compounds that are clearly having significant effects on the body. Part of what makes cannabis amazing is, to me, not so much the so-called psychotropic or psychomimetic effect, commonly referred to as the high, but that it has all of these other effects. And so if you look at the course of or strategy, the research strategy applied to cannabis from, let's say, the 60s forward, and maybe even further back than that, but definitely the modern era that led to the discovery of the endocannabinoid system was focused on cannabis as contraband drug material, okay? Illicit drug material, party drug, people might call it now. So, just to recap briefly, the work of Dr. Mishulam, and to his credit, he was able to do work on cannabis at all. So it's not slighting his work or his approach, but he was limited to using cannabis that had been confiscated by the Israeli police, okay? So that material, referred to as hashish, was completely uncharacterized, meaning how was it processed, what strains were involved, how old was it, what were just the general characteristics of it. So he really studied hashish. He didn't study cannabis. He studied a cannabis product, kind of like if somebody gave you a cigarette, you could study the effect of cigarettes. But if you say the effect of cigarettes is because of tobacco, that's a huge jump that shouldn't be made. But because people tend to ignore the parent plant, the botany, and I, like Jake says that he's a plant person, so I've been in this natural product space since I began, in chemistry, because back in the 70s, natural products chemistry was the great resource of new drug molecules. It wasn't because we hugged trees or anything. I mean, that's fine too, but there wasn't this, philosophical divide like people try to put forth now. So here's this space where, tobacco, sugarcane, opium poppy, coca leaves, they're not really toxic. It's only when we process them that they become something that can become contraband, that becomes a party drug, become a lethal drug, whatever. But nobody thinks that about cannabis, but it's exactly the same thing with cannabis. So hashish is dried material. What they had was not fresh plant material. And so what they discovered, first CBD and then later THC, structural elucidation, they spread the THC around with their friends, found out that they had a characteristic reaction that they associate with hashish. And so then THC is seen as the potent active principal molecule of cannabis. Based on what? Because it's in the highest quantity in their material sample, along with CBD. But all the other cannabinoids that are discovered later are then called minor cannabinoids. Why? Because they may have a 10 or 100 or 1,000-fold less concentration in hashish. Well, that really has nothing to do with the plant. And it doesn't even mean that they're not more potent. It just means they're more difficult to work with, because to get a kilogram of something that's in microgram quantities in your plant source, that's going to take a lot of time and money. So just to bring that along then, because THC caused this psychotomimetic, as they called it, effect that we call a high, then that's seen as the principal compound. So then that's carried forward into the 80s and 90s in the progression of the research as, well, how does THC work in the body? That leads to the discovery of receptor sites, that because there's an assumption that the primary function of cannabis is to get people high, then CB1, the first cannabinoid receptor discovered, and CB2, another cannabinoid, second cannabinoid receptor discovered, are associated with THC because of the association of cannabis with getting high. And so then that pushes everything over in this direction without realizing, well, what about the whole spectrum of CBD, minor cannabinoids, et cetera, which is what Philip is referring to, and why I wanted to tie it in here, that we're kind of figuring this out, you know, in the back end. It's almost like archaeology in Egypt. People are digging around. You know, we think that chemistry and pharmacology will be very straightforward. You start with the known and you branch out into the unknown and you characterize. But this has been like, oh, we were digging over there and we found this. And because we dug in this direction, we believe this is who the Egyptians were. This is what cannabis is. And, you know, we're a little spot in history, in the long, long history of the human cannabis relationship. So just as in closing, if Meshulam had worked with fresh cannabis, like Dr. Marilyn Barrett did in the 1980s, when she was doing her PhD in pharmacognosy, Dr. Barrett, in 1985, published the first paper that was the discovery and characterization of what are called canflavins, flavonoid molecules in cannabis that actually have have been shown to have a higher anti-inflammatory effect than non-steroidal anti-inflammatory drugs, and also than CBD. So, but what did her group do? Why did I bring it up? Because since they were pharmacognosists, they grew their own cannabis. They started with a seed and grew plants and characterized it, and then proceeded with standard extractions and then did assays. So she studied cannabis, but Shulam studied hashish.
Wow. Let me unpack a couple things here, because as when we always get together, there are so many ways that we could go. First thing is just for our audience, which I think will be very varied here. Thoughts on the canonical idea, I think one of you gentlemen had said, which is kind of like the standard thinking. Classically, we are trained a lot to think of pharmaceutical compounds in the model of a water-soluble IV drug. With a lot of the cannabis compounds, we're dealing with lipids, with fats, and they are just so stickier and fuzzier. Now, that's one pathway we could go down. The other pathway that I want to go down has to do with this concept, because there were some pretty bold statements here that I mostly agree with in terms of the processing of natural plant compounds. Let's start with caffeine, which is one that we all know and are familiar with. And the tea leaves naturally just picked right off of the plant. The caffeine is also accompanied with a secondary molecule, L-theanine. For many of us, myself included, who have had a little too much caffeine from time to time, we can get really jittery and even some anxiousness. But when L-theanine and the caffeine combine, that has an effect of leveling out the negative consequences of the caffeine. So I wouldn't call that an example of synergy, but I would call that at least an example of cooperation. When we see with cocaine, which is a purified derivative of the coca leaf, if you go to Bolivia, Colombia, you'll see the local peoples that are working hard every day to chew the coca leaves. I have had, out of this country, I can say, had the opportunity to have coca tea. It was delicious.
Right.
I got a very clean sensation out of it. So, but as you purify the molecule from the coca leaf to powdered cocaine, you have one effect, and an even more, what is an even more purified, and I know I'm shouting here, but an even more purified form than powdered cocaine is crack. And we know that, so same is true with sugar. Raw sugar cane versus raw unrefined sugar crystals versus white table sugar. Huge differences in rates of cavities depending on the amount of white sugar that is consumed versus closer to the natural. So that's one concept, and then the other is this.
And white sugar is psychoactive.
Oh, every definition I can think of a drug, white sugar fits. Can you think of an example, a definition of a drug that white sugar doesn't? Lands on receptors, creates a euphoric effect. There's a resultant crash. people can become addicted to sugars. But anyway, I'm going to step back. But remember, if that's brought anything up for you, and also the water soluble versus the lipid soluble, and we haven't heard from.
Well, no, Jake, we need to know why you're here.
Why am I here? I
Yeah.
feel that one of the reasons, I mean, philosophically on the planet, I don't know, but one of the reasons that I think I'm here is because I am trained as a generalist. I have background in pharmacology, biochemistry, et cetera, but I'm not a specialist. And what I see with the endocannabinoid system is kind of the re-rising of the generalist in terms of efficacy and understanding. Because if you are focused on, if you are just focused on renal tubules, you are missing a part. I mean, as a specialist, as a nephrologist, you're missing a part of the big picture. And part of what we need to understand with the endocannabinoid system is that it's not only working in small areas of the nucleus with PPAR gamma, et cetera, but it's also working globally and arguably even on a societal level.
Yeah, and so part of, you know, what's going on is when you have a fresh plant, then you have another array of compounds. So to me, like the biggest nail in the coffin, if it's not several nails in the coffin, is that THC and CBD are artificial compounds, meaning they're not natural products of the plant.
Explain that a little bit for folks.
Okay, so in the plant itself, in the cannabis plant, the parent compound, for instance, cannabidiol, cannabidurolic acid, is produced by the plant.
For everybody's.
Yeah, CBGA, okay, and it's called gerolic because it generates these other compounds. So the way that, it's kind of like a family tree sort of a thing. This compound begets this compound and that one and that sort of a thing. And so this parent compound, depending on which enzyme it's acted on within the cannabis plant, either produces tetrahydrocannabinolic acid or THC acid, the acidic form of THC, or the acidic form of CBD, cannabidiolic acid, okay? So that's what's present in the plants as the end of that pathway, okay? It's not until those molecules kind of like leave the nest, until they are taken away from the living plant, until the plant is cut down and dried, or if the plant is grown under such conditions where there are either stresses, environmental stresses, including, say, poor cultivation, and you allow it to dry, out there on the land of outdoor grow, perhaps, then you start to have oxidation. Usually people refer to it as decarboxylation, but you tend to have a degradation, let's say, of these acidic forms into the common forms that people associate with cannabis because of the work on cannabis being with contraband, okay, being with hashish, and even prior to that being processed forms of cannabis as opposed to, kind of a canonical, if you will, again, use that word, approach to the rational analysis of the pharmacologic effects of the cannabis itself. So it's not until, for instance, typically cannabis is dried and cured, and then in the recreational, as it's called, context, smoked. So that exposes these acidic cannabinoids to high temperatures and also to ambient air oxygen that then causes their degradation from the acidic forms to the familiar forms of THC and CBD. If one does not decarboxylate, say, freshly cut cannabis flower, even if it's completely so-called recreational grow, OG Kush, whatever the different strains, it's not going to have THC. It doesn't have THC until
THC.
Pardon?
It will have, I'm just repeating for the audience. It will have THCA, but not yet THC.
It will have THCA. Unfortunately, because of the bias towards cannabis as marijuana as a recreational drug, then the regulations dump all of the variations of THC, including THCA, into the same box when you look on, say, a label, here in Washington, California, wherever, what's the THC content of this product, say at a dispensary, it gives total THC, and that includes the THCA. And if it's something that has not been decarboxylated, often that THCA level is higher than the THC. So anyway, the point is that THCA does not get people high. It also is not addictive, but THC isn't addictive either. It also does not, according to the literature, does not get converted to THC in the human body, because of course the human body is not going to reach temperatures equivalent to smoking or the high temperature processing. That would trigger the conversion of THCA into THC. It has its own metabolic pathway within the body as far as its elimination. But again, going back to the regulatory standards, when somebody takes a urine test, the presence of THCA metabolites have long since the 70s and 60s been considered a sign of cannabis use. So it's still put in that demonized category that, oh, this is an irresponsible pothead, which is just part of, non-scientific folklore, agency folklore, the National Institute for Drug Abuse, NIDA sort of folklore, because you don't actually know why the person was using the product. And you also don't know if they ever got high, because if they use a little bit every hour, every day, they could reach the same levels as somebody who went to a Grateful Dead concert for one day, as far as the metabolite. So there's a lot of pseudoscience in there that has become policy. But the reason I wanted to mention it here is because CBDA and THCA, for instance, CBDA has been shown to have 1,000 times the affinity for the 5-HT1A receptor, serotonin receptor, that's associated with reducing nausea and vomiting, and also is associated with modulation of mood. 1,000 times is a big deal. That's not a minor cannabinoid. I'm saying 1,000 times more than CBD.
And CBD.
Okay.
So before the molecule, before it is altered by us in the manufacturing process or in the combustion process, has a higher affinity for the serotonin receptor. I'm gonna I don't mean to interrupt. I want to ask Philip in regards to some of these compounds, THCA or THC even, as lipid-based receptors. How do you see the status of the science moving forward? And if you could tie one of the cannabinoids landing on the PPAR receptors into that thread, that would be wonderful.
Well, I'm not sure I can do that. My focus has been at the CBD level, but I've been looking at the other endocannabinoids like palmitoylethanolamide, oleoethanolamide, which are alpha PPAR receptor agonists, and they orchestrate a number of different other subtleties and improvements on the body, as well. Well, with CBD, what you have is you're working on the PPAR gamma receptor, which is that's that cascade that goes along with that. But part of those other endocannabinoids are interacting with these PPAR receptors in a very important way.
In terms of dietary, I have heard some folks speculating that as the body ages, supplementation with cannabinoid acids becomes more conditionally, or can be more conditionally helpful in the aging physiology of humans. How do you see any differences between, just in thinking of overall, taking these things into our bodies? We don't know exactly what's going on, but do you see the aging process and any of these lipids, PEA that you were discussing, or even omega-3s becoming more and more necessary as the physiology progresses towards senescence?
Well, I don't think it's beyond what we already know is a healthy diet, and these are healthy substances that we bring in. Much of the work that you've been doing already, and you've already recognized that omega-3s have a particular role, and that limitation of refined foods is a very, very much of a problem. And we're orchestrating a number of these changes, and these are the signaling mechanisms that are actually inducing many of these changes. With respect to longevity, yeah, I'm very interested in longevity since I turned 70 in a couple of weeks. Survival mode.
Yes.
Now, how can I maintain myself at optimum performance and reduce the effects of aging? And I think the cannabinoids, yes, they have an effect, but we also, I think, are learning about and we have a better understanding of some of the similar targets of the cannabinoids and how they work now within the body. And so we do know that CBD actually improves longevity. That we do know that beta-caryophyllene found in many of the foods that we take in, whether it's black pepper, oregano, basil. I mean, that has a very specific, and it's been shown to increase the longevity of the elegans by 22%. So these are impressive statistics, and they really give towards the concept that these are healthy for us. But are they more healthy than the natural substances and the natural cycling, let's say fasting and then feeding and nutrient compounds that we bring in? I'm not so sure they are. Finding out what the right pattern is, and do we need a boost when we get depleted at some point in time? For instance, ALA is alpha linoleic acid is a great substance and is an omega-3, but it can't be processed as efficiently in the older age group. So then you might need to be taking in some fish oil or EPA or DHA
or hemp seed oil
to affect those benefits.
Or hemp seed oil as well. So
yes.
It's interesting to me. Go ahead.
Well, then, I don't believe that hemp seed contains EPA or DHA.
No, I'm saying that that in addition. Go ahead.
I should know that, and I don't. Hemp seed oil does have anti-inflammatory omega-3s, and it also has, I believe it's the GLA, which is an anti-inflammatory omega-6. So I do think of it as an and also not only does it have a good lipid profile, but it has a perfect protein profile. And I always think of, I mean, not to totally sidetrack it, but as humans, many of us are thinking about Elon Musk, for example, colonies on the moon and Mars. If we bring these, we're going to have to grow something up there. If we bring these seeds up there, it's just the most wonderful food source. And just to bring things more back to the more practical, the hemp plant or the cannabis plant is so easy to grow outdoors. I've heard it argued that we could feed the entire planet with a hemp seed diet, and then it would be beneficial. So anyway, sorry to interrupt.
Well, here there's I just did a little quick Google search, and there's an article here from Paul Ben Haim, who I believe is the head of the Elixinol company, but he mentions in here that that DHA and EPA are part of hemp seed oil. But I'll just I don't have a regular article on it.
Did you say THA? Sorry, you meant
DHA.
DHA. Okay.
Yeah. So anyway, among the different compounds, let's see. Nope, that one's the next one down. Anyway, just the point is, though, that hemp seed oil can serve as nutrition, if you will, for the endocannabinoid system, leading to increased production of endocannabinoids. The acidic cannabinoids, for instance, THCA and CBDA, it's been shown, there's a study that was done, I think, at Stanford, pharmacokinetic study, that two different studies actually, that both showed that cannabis oil, cannabis extract products that contained acidic cannabinoids versus the ones that were fully decarboxylated, where the acidic cannabinoids THCA and CBDA were both present, there was actually a higher bioavailability of CBD and THC. Meaning in those products, there was a presence of both the acidic original parent cannabinoids, the acidic forms, and also the decarboxylated forms because of whatever sort of processing and storage that product went through. But as a result, in assay, you see both the THCA and THC, CBDA and CBD. But when you have the presence of THCA along with THC, when you have the presence of CBDA along with CBD in that same product, you actually have an increased absorption of CBD and THC. I mentioned that because we, in this modern culture, the concept of panacea, cure-all is anathema. Nobody likes to think that we can get all of the answers from one place. But cannabis comes pretty close. Between the seed, hemp seed oil, and the flower, between the acidic cannabinoids and the decarboxylated. And just so that nobody gets it wrong, I'm very happy that they discovered the decarboxylated form, because CBD and THC have some bioactivity that you don't see with CBDA and THCA. For instance, THCA, it's not even clear that it binds to CB1 receptor at all. So there's some claim that it binds weakly, others say that it doesn't bind at all. But to imagine that there's a THC molecule that doesn't interact with the CB1 supposed get high receptor is like a breakthrough concept to even consider. So there is this synergy, for instance, in the context of anti-inflammatory, from canflavin A and B, the unique flavonoids that are found only in cannabis plants that I mentioned from the work of Dr. Barrett back in the 80s. Then you have CBDA and THCA that also work as anti-inflammatories and can work synergistically with CBD and THC from the standpoint of pharmacokinetics, but also because the canflavins also hit, for instance, 5-lipoxygenase, whereas the acidic cannabinoids, you see cyclooxygenase activity and some specificity for cyclooxygenase 1 and even cyclooxygenase 2, which is the golden target of non-steroidal anti-inflammatory drugs, but without the associated issues with platelet aggregation, with GI irritation and bleeding and all those things. So that's a big space. It's a very important therapeutic space, and as Philip mentioned, it's a very important space with respect to longevity, not just arthritis, but in multiple contexts, because inflammation in the CNS can lead to depression, and in the GI tract, you have malabsorption, and then you also can have a loss of serotonin, which feeds back into depression emotionally. Then also there's a lot of implication of inflammation and the carcinogenic process, which as you add decades onto life, this is where I started in undergrad and medicinal chemistry, was looking at the process of what's called tumor promotion. And so most people don't realize that breast cancer doesn't show up after you have a fun weekend in Vegas. It's 15, 20 years that it takes from the first cell that becomes malignant until it grows large enough to be detected and then therefore be diagnosed as a disease. Colon cancer, the estimates are 40, 50 years. So the space I was looking at, tumor promoting agents, the receptor site for the tumor promoting agents that I was looking at, for instance, the 4-ball esters that also come from a plant from India, they were found to bind to what's called protein kinase C. Well, protein kinase C is right next door to the cannabinoid receptor. And actually the action of tumor promoting agents affects the CB1 receptor, the cannabinoid receptor, it can cause phosphorylation, which then changes its bioactivity in the cell. So all of this stuff is connected. For me, it's like some sort of medicinal organic chemistry karma for me, that no matter how many different directions I've tried to go in my research career, it still comes back to the same space. When I was doing tumor promoting agents, there was protein kinase C had not been discovered yet. The endocannabinoid system hadn't been discovered yet. So there was no way to connect those dots. But sitting back as not as old of a man as Philip, but older than you, Jake, you know how you sit there and look out the window and you rethink over your life? Some people think of their conquests, brag about their heroism in war, or women they met on the beach. I'm thinking about molecules that I met over the years. And it's like, oh my God.
Where's the duct tape? Where's the duct tape? Hey, I've got a concept for you. Now, I've been thinking about adaptogens, and you mentioned adaptogens earlier, Jake. And one of the ideas there is, what is an adaptogen? And what I want, and you can explain that, but I was, and Calumet, you brought it to mind as you talked about these different connections that you're making all over the place. As I explore the endocannabinoid system, I am finding, I tend not to use an agonist or as an antagonist very early on, because there's a modulation that occurs. And so with the adaptogenic idea is that we've got these mechanisms in all over the place, every part of our body, including the interstitial system, that's your favorite. But it behaves differently and acts differently depending on the tissue, the state of the tissue, and the environment that's surrounding it, and that particular grouping of tissue, like a cell group within that particular tissue. And as a result of that, where you throw out a cannabinoid into one of those areas, or endocannabinoid, you get different responses in different areas. And to me, that says, oh my God, that is an adaptogen. Something that is going to have different effects in different parts of the body, generally beneficial, but that diversity is sort of cascaded out there. What do you think?
Well, I like that a lot, and it's true. And when you get into the details, it can be very confusing. For example, a certain ligand might stimulate the endocannabinoid system in healthy tissue, but if the tissue is diseased, it might have the opposite effect. I think of an adaptogen as that anything that assists the physiology to adapt to any type of change, because the outer world is we don't have any, as a biologic organism, we don't have any control over we have little control over the outer world and how it affects us. So the endocannabinoid system developed 600 million years ago from single-celled organisms that got together to form bodies. And what is the first tissue? There are 4 types of tissue. The first tissue that developed was connective tissue. It's a communication medium. And so this is why I think the embryology is very important to and that's an area that I strive to understand better, Lord have mercy. But the embryology is very important because as the kind of debunked saying goes, ontology recapitulates phylogeny. As the history of life evolved from simple multicellular organisms to organisms with tissues to, small neural nets toward the mouth and et cetera, coordination systems evolved. And they're typically between a water and a lipid interface. The and I guess all signaling is doing that. So the system, the biologic systems had to solve a certain number of problems. One is how do I get simple undifferentiated tissue to become differentiated? What's the pattern of growth in the organism? That's one problem, and the ECS does all of that. The second problem that needed to be solved is what if there is an injury to the organism? How do we even detect that this part of the body is damaged? And then how do we manage the biologic resources necessary to do that? The third area, energy metabolism. If you're bringing cells inside of a body, they don't have access to the primordial seawater, so we got to get food to them and we've got to get waste products out. Again, the ECS, which I don't even think, I think of it as more of a lipid signaling system than a cannabinoid system. The biology needs to respond to all of these problems, both globally and specifically. And this is why I'm an interstitial guy, is because that is from the very beginning the medium of exchange of information. For example, the synapses in our brain, where literally nerve cells are communicating with each other. That little tiny area is interstitial space. And it fascinates me. So an adaptogen helps the body adapt, and it does that by assisting or enabling the organism to detect and respond to change. It's a very general concept.
Yeah, it's a general concept, and you explained it wonderfully. It's just so that people don't associate general with vague or unimportant. It's essential because, you know, especially now during this time that we're, you know, recording, being a time of, you know, returning to quarantine because of, you know, coronavirus concerns, the concept of what happens when we move into different environments is of paramount importance, because these devices that we live in, called human bodies, are designed to spontaneously adapt to changes in the environment. And in the age that we live in, say with airline travel, and even faster with some of these SpaceX devices to be able to travel any place in the world in an hour, suddenly the body can go from being in Antarctica to being in the Sahara Desert. And you don't want this device, this smart device called human body, to fail. It needs to continue to function. And so this adaptability is essential in the ability for the functionality of human physiology to continue to work regardless of the environment that it's in, if it's underwater, if it's at elevation on a mountain, all those sorts of things. So that's at least as important as everything else that's going on in the body, because if the physiology doesn't adapt, it doesn't matter how healthy one's heart or brain or muscles or other vital organs are, because the way they interact and the way they communicate, as you're mentioning, Jake, is essential because everything's context.
And I think that the concept of adaptability and adaptogenesis or adaptogenic processes, for me, CardioMed accused me of being a young guy. I remember when I was 36, I said to my dad, "Boy, Dad, I really feel like I'm getting old." And he laughed. He said, "Oh man, what I would do to be back in my 36-year-old body." And for me now, 36 seems so very young. But the point is that as senescence occurs, as the organism ages and our biologic machinery becomes less and less efficient, these properties that help coordinate crosstalk between different cells, different tissues, different organ systems become more and more important, not only just for survival, but for a good, vibrant life with more greater numbers of good days. So anyway.
Every day is a good day with you, Jake. I've never had a bad day with you.
That we definitely want to collect those good days and have them going forward versus the negativity that we've seen more recently in our environment and on the air.
Yes. Yeah, most definitely. And the thing is, it's like sometimes people talk about body awareness. There's nothing like aging to give you enhanced body awareness, because, you know, when you're in your teens or your early 20s and you pop out of bed and you have a couple of roommates and maybe you have a pillow fight and wrestle, and then you go mountain biking down somewhere and then eat a bunch of crazy food, pizzas and burgers, and then have some beers and fall out for the afternoon. And then tomorrow you feel fine and you can do it again. it's kind of, you don't realize that there's a human body being in there. And part of aging to me is reminding myself, because I used to play a lot of sports, that it's in my mind, at least, it's like old injuries never go away. They just go into hiding. 20 years later, after that head over head tumble from snowboarding, mountain biking, bicycle kick, and soccer, whatever, then suddenly you remember, it's like, oh, because suddenly the whole constellation of it, they call it body memory, some people in the kind of, alternative medicine. Oh, that's why my wrist, neck, back, and you wake up this morning as though you did it yesterday, and it's like, oh, thank God for CBD.
and that, I think, ties in with the how do we dose cannabinoid products. It's not only on an individual basis, but it can very much be on a day-to-day basis. And again, it can be a teacher listening to my body. How am I doing? I'm feeling pretty good today. I did my morning stretching, and that.
What a great topic! I think we can do the next hour on that. I think this is I think would be a good time to wrap it up for today.
Hey,
that we talk about aging and we dismiss aging. And the reminders, and then we pick up on, let's talk about dosing next time that we get together.
It's so great to see both of you guys. You guys are just really, I have the utmost respect for you. I learn from you all the time. Thanks for being here.
Sure. Thanks for inviting us, Jake. And just so that in the name interest of transparency, I want the people that are watching this to realize, even though this is a Zoom call, we're actually all our roommates. We live in the same house.
These
are just separate. We're actually sitting about 6 feet apart from each other, but we have these virtual backgrounds to make it look like we're spanning the globe, because it makes us feel important.
I do. I feel important. Gentlemen, great to see you.
All right. Thanks.
Great. Bye.
Bye-bye.
Walk with me as we take a broad-ranging tour of the world of cannabis, from the ancient to the modern, the medical to the recreational, as well as the social, cultural, and political landscapes of this fascinating plant. I am Dr. Jake Felice, and this is the Cannabis Matrix, where cannabis science meets cannabis culture, where recreational meets medical, and where the body's own endocannabinoid system is as much a player as CBD or THC. Welcome.